Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speaker, Volkmar Tom, PhD, Head of PD Separation Materials from Sartorius.
Thank you for this kind introduction. Hello to everybody online, and thank you for joining this webinar. I'm very happy to introduce you to a new membrane technology that we have developed over the last years here at Sartorius that we named Convecdiff. This new Convecdiff membrane platform is designed for intensified capture of monoclonal antibodies. In this webinar, you will learn why we developed this technology and what advantages it offers compared to traditional chromatographic resin or purely convective materials that already exist. We will start with a short background and motivation, followed by the goal we wanted to achieve with this technology development. We will then introduce you to the novel Convecdiff membrane platform. We will show you the chromatographic performance of a protein A affinity membrane based on the Convecdiff membrane platform, as well as the respective scalable device platform. Today, protein A affinity capture chromatography is almost exclusively performed by the use of chromatographic resin particles packed in a column. There are different particle materials available. However, these resins typically exhibit a bead particle diameter of around 50 to 100 micron. Antibodies will need to diffuse into these particles in order to utilize their binding capacity. Due to the relatively large hydrodynamic diameter of antibodies of around 10 nanometers and the resulting low diffusion coefficient, the diffusive path length of 25 to 50 micron represents a considerable mass transfer resistance, making long residence times necessary to achieve a reasonable level of dynamic binding capacity. On the right-hand side of this slide, you can see two typical examples of commercial resins and their dynamic binding behavior as a function of residence time. These resins would typically be used at a residence time of 4 to 6 minutes. These long residence times lead to a low productivity of only 10 to 20 grams of antibody per liter of resin and hour of processing. A full chromatographic cycle takes hours of processing time. Therefore, only a handful of cycles can be performed for a single batch. After each batch, the packed column needs to be sanitized. Cleaning validation becomes a necessity and bioburden risks need to be managed. Last but not least, large capital expenditure need to be made. An additional pain point is the underutilization of the resin lifetime during clinical phase and related costs. Our goal was to enable rapid cycling chromatography with cycle times of minutes rather than hours, leading to 10 to 20 times higher productivity compared to packed bed resin technologies, leading to full lifetime utilization within a single batch. Rapid cycling chromatography needs three prerequisites: a chromatography membrane with higher mass transfer rates and high binding capacity. Second, a scalable device portfolio. And third, a rapid cycling chromatography system with low holdup volume and rapid valve action. Today, we will focus on the membrane and the device portfolio in which it will be available. It is obvious that purely convective materials like fibers, monoliths, and convective membranes can have extremely fast mass transfer characteristics, as the transport to the binding sites is predominantly achieved by the convective flux through the material. The schematic on the right-hand side illustrates the mass transfer situation with affinity ligands immobilized on the pore wall of these convective materials being accessible by convective transport. This mass transfer consequently leads to a dynamic binding behavior that is almost independent of the applied residence time. This property can be seen on the next slide, where the residence time dependency of the dynamic binding capacity for a fiber-based material is shown in direct comparison to the residence time behavior of common protein A affinity resins, as already shown in an earlier slide. The data underscores the very fast mass transfer that convective materials exhibit. However, for these convective materials, binding capacity scales directly with the amount of inner surface area that is accessible to convective flow. In this slide, we can see the trade-off between the nominal pore size on the x-axis of the respective convective material and the resulting binding capacity on the y-axis when functionalized with a protein A affinity ligand. Independent of the material type, may it be membrane, fibrous, or monoliths, we can see the expected tendency. In order to achieve higher binding capacity, the material will need to exhibit a reduced nominal pore size in order to increase the specific inner surface area of the material. In order to achieve a reasonable binding capacity of, let's say, 35 grams per liter, the material will exhibit a nominal pore size of about 0.3 micron, is to say a pore size close to the pore size of a sterile filter. Such small convective pores will, however, lead to issues. The resulting low permeability will lead to elevated pressure drops, requiring low bed heights in order to enable short residence times, making it difficult to design a scalable device family. Furthermore, small convective pores introduce a high fouling propensity, making it difficult to achieve reproducible and robust processing over many cycles. The novel convective membrane platform brings together the positive characteristic of both technologies, is to say, fast mass transfer and high binding capacity without exhibiting small convective pore sizes. This is achieved through the introduction of a structured hydrogel that represents the binding phase, very similar to the binding phase in traditional resin beads. However, this hydrogel phase has a very short diffusional path length of about 3 micron, making fast diffusive mass transfer possible. The pore size of the convective pores is therefore uncoupled from the binding capacity and have diameters of around 5 micron, leading to high permeability and low fouling. This novel structure enables high productivity and scalable and robust performance at the same time. On the left, you can see a confocal laser scanning image of the membrane where the green color depicts the binding hydrogel phase and the black color the convective phase. Both phases represent about 50% of the material volume. You can see the very regular bicontinuous structure leading to efficient convective mass transfer and a very narrow distribution of diffusive path length. On the right-hand side, you can see an image of the structured hydrogel taken by cryoscanning electron microscopy. Pore sizes of the hydrogel are in the range of 100 nanometers. From this illustration, it becomes obvious how far this novel convective material differentiates itself from purely convective materials. The dynamic binding capacity and pore size are not coupled anymore, enabling high binding capacity even for large convective pores. Integrated into a family of self-contained capsules and modular cassettes, the membrane bed will have a constant bed height of 4 millimeters over the entire range of scalable products from 1 milliliter up to 24-liter bed volume. For this format, you can see the dynamic binding capacity for a panel of monoclonal antibodies and Fc fusion proteins ranging from 30 to 58 grams per liter at a residence time of 12 seconds. Benchmarking the dynamic binding capacity at 10% breakthrough as a function of residence time to state-of-the-art resins and fibrous convective material, the performance advantages become apparent. The new convective material exhibits high binding capacity already at short residence times. In addition, dynamic binding capacity can be modulated to even higher values when the residence time is increased, similar to the behavior of resin beads. When benchmarking the critical performance characteristics to a traditional protein A affinity resin using the same feed, we can see that overall yield as well as the purity of the eluate are very similar, while productivity is increased by a factor of 15 to over 200 grams per liter and hour. Many cycling studies have been performed in-house and at external collaboration partners with this material. This slide only shows one of them in order to underline that this material is capable of performing many bind and elute cycles. The UV as well as the pressure traces show no variation or trend over all cycles. The caustic stability of the membrane and the ligand are sufficient to perform a 0.2 molar caustic CIP step at every cycle. Contaminant clearance levels are constant over all 200 cycles. We want to highlight that even though equilibration and washing was executed at 10 membrane volumes per minute, is to say at a residence time of 6 seconds, the pressure drop never exceeded 1.2 bars. Let us now have a look at the second pillar that enables rapid cycling chromatography, which is the device portfolio. We have integrated this new membrane in 2 different screening devices, namely 96 well plates as well as the Pico devices exhibiting 80 microliters of membrane, as well as into a scalable device portfolio from the 1.2 milliliter nano up to cassette assemblies for 10 or more liters. I want to point out the constant bed height over the entire range of scalable products. The scalability of this device portfolio has been shown for the existing Cytobind membrane portfolio already, namely for Cytobind Q and others. Here we show data that confirms the scalability for a bed volume of 1.2 to 75 milliliters, also for the novel Convecdiff protein A membrane. Further scalability for 10-inch and cassette assemblies will be available soon. Conclusions. Due to inherent structural characteristics, Convecdiff membranes offer unique possibilities in the area of ultra-fast protein capture chromatography. The high volume gel phase ensures high binding capacity, while at the same time, large convective pores of more than 5 micron ensure robust cycling action at a minimal CIP effort, device scalability due to constant bed height over the entire device portfolio, and a high permeability for low pressure operation. The short diffusional path length of less than 3 micron ensures high mass transfer rates and high productivity, typically a factor of 10 to 20. This high productivity enables the concept of single batch use as well as accelerated speed to clinic. And the availability of modular cassettes enables the scaling to large production processes. I want to thank you all for your interest in this new technology. We hope that we could explain why and how this new technology differentiates itself from existing alternatives in the market, namely resin technologies and purely convective materials. Please contact us with any questions you might have or ask your questions directly in the live Q&A session. Thank you. I don't hear you. I don't hear you, Alex. Do you hear me?
Hear me now?
Yes.
Okay, sorry about that, everyone. It seems my microphone decided to shut off. So, as I was saying, as a reminder to our audience, you can go ahead and type in your questions for our speaker now. Our first question is, do you have also such membranes which are grafted with DEAE molecules?
Yeah, this is a good question. The first product that we just launched now is with an affinity ligand, a protein A affinity ligand. This platform is capable of other functionalities. We have tried that in the lab. They're not available yet commercially, but that is a possibility that we are looking into, and more functionalities will follow on this platform, I'm pretty sure.
Okay, and how is sanitation in CIP typically performed?
So the membrane is caustic stable to a degree that enables hundreds of cycles during protein capture. So typically in the beginning, we recommend half an hour of 0.5 molar sanitization. It's certainly the choice of the customer to do that or not, but this is what we recommend. And then the CIP step is typically 1 minute at 0.1 or 0.2 molar caustic during each cycle. This is our recommendation. We have seen that you can run this membrane also for many cycles without CIP, but it really depends also on the feed stream and the properties of the titer and so on. So it's, when you want to have a robust process, it probably makes sense to do a CIP step at each cycle, but it's not necessarily necessary. You can run 50 to 100 cycles without that potentially. No, it's something you have to try out.
And how many cycles can be performed without losing performance when material is clarified by centrifugation or sterile filtration instead of depth filtration?
So this we have not tried. I have to say we have always done a typical centrifugation step followed by a very typical normal depth filtration step. You can read that in our publications that are coming out. And there we have run hundreds of cycles. As you see in the presentation, 200 is no problem. We have run more than that. So it's very robust. However, not using a depth filter is something that we have not tried so far. So that's an interesting question that I cannot answer really. Yeah.
And is a pre-filter needed in front of the Rapid A membrane?
We certainly recommend it. I guess it's nice to have a police filter in front of a chromatographic material always. So we would strongly recommend it. The same here, it might not be necessary always, but it's probably a good idea to do that as a police filter and for robustness sake.
And are there any studies done on input feed concentrations, impact of higher titers on DBC?
Yes, so we typically don't see more or less impact than you see on a chromatographic resin that's in the market. There might sometimes be impact. If there is, it's in a similar range. We have seen very nice data for example, very low titers, where you can save a lot of time and gain productivity because of the fast capture step that you can perform with this membrane compared to a resin. There is no pronounced relationship here between titer and dynamic binding capacity that we have seen.
And do we need a separate system to work with this membrane, or can it be used with our traditional FPLC systems as well?
So we have designed a multi-use chrom system that is especially designed for membrane chromatography. The difference is that you have low hold-up volumes and at the same time have high flow rates. And this is also accompanied by very large, very fast valve action. So those components make for a low dispersive system that is very advantageous for this kind of operation. However, you can use any chromatography system that generates the right flow rate. The only drawback is that a typical resin chromatography system that creates these kind of flow rates has typically also large dead volumes. And you know that the membrane that you use is much smaller than a resin column. And then the ratio between dead volume and column volume or membrane volume becomes, a little bit disadvantageous for the dispersion. So it's not recommended to use a traditional chromatography system, but it can be done.
So it looks like we're running a little bit short on time. So if we don't get to your questions, we'll be passing them along to Volkmar, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, how does this material compare to the Gore protein A membrane?
That's a good question. So I've not discussed this material in my presentation. I've discussed traditional resins as well as convective materials, fiber-based or membrane-based. The Gore protein A membrane that is also coming on the market is a membrane that has some characteristics that we share. It has a diffusive phase, just like our membrane. However, it still has similar quick mass transfer. There are some differences, though. As I explained, our membrane is made from a very hydrophilic hydrogel that has very low unspecific binding and is a wonderful material for chromatographic processes. So the binding really only happens at the affinity ligand or at the functionality that you would introduce into that system. My understanding is that the Gore membrane has a synthetic hydrophobic polymer as the base matrix, where the diffusive phase is embedded into, and this might lead to adsorption and fouling, and this might be a major difference here to our system. Now I lost your voice. I don't hear you. You need to have a new headset, Alex. Yeah, we don't hear you.
Can you hear me now?
Yes.
Oh,
now it works again.
I'm sorry, everyone. I guess I do need a new headset.
Oh, yes.
Well, as I was saying, thank you. And the recorded version of this webcast will be made available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Goodbye. Thank you, Alex. Goodbye to everyone.